So today's topic is a Unit 1 subtopic buffer. So this rectangular shape is represented by the biological system, for example human body. In the human body there is lot of chemical reactions happens, lot of reactions happenings, enzymatic reactions are happenings. So all these chemical reactions happens at a specific pH. So every enzymatic reaction occurs at an optimum pH. Clear? Metabolism. The biological system. In the biological system, metabolic activities are more regular. So when there is metabolism, more acids and more bases are created. So when the acids are added to the system into the tissue fluid or blood, or the base is added, it alters the pH. Clear? So when the acids are produced, that means more H plus ions are added into the system. That means the pH decreases. When the pH decreases, that means it becomes more acidic. Similarly, if it is bases is added, that means more OH minus ion is added to the system. That means the pH increases or it increases the basicity. So increase in acidic condition or basic condition alters the pH, which affect the chemical reactions. So in order to perform the chemical reactions, normally the pH has to be maintained properly. And how does it happens? So this is maintained by a system called the buffer system. So what is a buffer? What is a buffer? Buffers is an aqueous solution. Aqueous solutions or an acid base balancing solution capable of resisting any pH change, or either decrease or increase any pH change of a solution when excess acids or bases is added on it. Clear? So once again, a buffer is an aqueous solution or an acid base balancing solution capable of resisting any pH change, decrease or increase when the acid or base is added to this system. Now how does it? So what is a buffer? So buffer consists of a weak acid and its conjugate base, or a weak base and its conjugate acid. So there are two things in a buffer. Either it's a combination of weak acid and its conjugate base, or it contains a weak base and its conjugate acid. So what is an example for weak acid and conjugate base? The examples are acetic acid is a weak acid and its conjugate base is what? Yes, a sodium acetate is the conjugate base. So this forms a buffer. One of the example for buffer. And the second example is if the buffer is a weak base and its conjugate acid, for example ammonium hydroxide and is a weak base, and ammonium chloride is its conjugate acid. So this is another example for a buffer. Got it? So there is another important terms called buffering capacity. Buffering capacity. So what is the buffering capacity? Is the capacity or the effectiveness of a buffer system to resist the fluctuation in pH. Fluctuation in pH is called a buffering capacity. So once again, buffering capacity or its effectiveness means is to resist fluctuation in pH. It's actually called the buffering capacity. So the capacity or the effectiveness of a buffer system to resist the fluctuation in pH is called the buffering capacity. So it depends on two factors. There is one factor is the concentration of conjugate base plus weak acid is concentration. The second one is the concentration of the buffer components. So these two factors affect the buffering capacity. So what are they? So one is the conjugate concentration of conjugate base plus its weak acids, and second one is the concentration of the buffer components. And what is this importance of this buffer? It maintains the physiological pH. The main function of the buffering system is it maintains the physiological pH of the cytoplasm and extracellular fluid. So what are the major buffers in the human body? So one is it's very important, phosphoric acid and phosphate, or it is called H2PO4 minus bar HPO4 2 minus. So this is a very important phosphate buffer system. And second one is what? Carbonic acid plus bicarbonate buffer system, represented as H2CO3 bar HCO3 minus. So once again, so these are the different examples of buffer system that is seen in the human body. So what is the first one? It's a combination of phosphoric acid and its phosphate, H2PO4 minus and HPO42 minus. Then second one is the carbonic acid plus its bicarbonate, and third one is the hemoglobin buffer system. ശ്രദ്ധിക്കണ്ടല്ലോ, നിങ്ങൾ മൊബൈൽ ഓൺ ആക്കിയിട്ട് ടി വി കണ്ടുകൊണ്ടിരിക്കുകയാണോ? So HB buffer system. So this is the three buffer system in the human body. Let's one by one each. Now let's look at the examples. So this H2CO3 is dissociated into H plus plus HCO3 minus. So this is one buffer system, H2CO3 HCO3 minus. So what happens when sodium hydroxide is added in the system? When the sodium hydroxide is added, what happens is split into what? Na plus plus OH minus. So when the OH minus is added into the system, that means it increases the basic condition. The pH increases. So that has to be brought to the normal. And how does it happens? So there is a way. So this addition of OH minus ion has to be neutralized. And how? This OH minus ion and this H plus ion combine together to form what? H2O. So this extra OH minus ion is the action of OH minus ion is neutralized by combining with H plus ion into water. Now these two combine together to form what? NaHCO3, sodium bicarbonate. So here the addition of an alkali into a system has been neutralized so that the pH is maintained. On the other hand, if the acid is added, so that liberate more H plus ion in the system. So what will happen? This H plus ion combined with HCO3, CO3 minus ion to form what? H2CO3, that gives rise to H2O plus CO2. So then net effect of H plus ion is neutralized and produce what? Ultimately the water and carbon dioxide is produced. So this way the pH is maintained normally. So this is called bicarbonate buffer system. Okay, last 2 minutes. ശ്രദ്ധിച്ചിരുന്നോളേ, ബോറടിക്കരുത്. Ready? So next one is what? Second buffer system is called the hemoglobin buffer system. So during metabolism, the carbon dioxide are formed in the tissues. This carbon dioxide combined with water in the blood. So when during metabolism, what happens? The carbon dioxide are produced. So from the cell, the carbon dioxide are diffused into the blood. So in the blood there is water, but RBC, inside the RBC there is water. So this carbon dioxide combined with water to form what? These are reversible reactions. H2CO3, which in turn dissociated into H plus plus HCO3 minus. Clear? So once again, so this carbon dioxide is produced inside the cell due to metabolism. So this carbon dioxide produced enters the bloodstream. From the blood it enters the RBC. So from there, carbon dioxide combined with water to form what? H2CO3, carbonic acid is formed, which dissociates into H plus plus HCO3 minus ion. So this H plus ion increases the blood pH, which is actually 7.4. So this should be maintained at 7.4. And how does it take place? So this H plus ion combined with the hemoglobin to form HHB. This is called hemoglobinic acid. Globinic acid. So this extra effect of H plus ions is reduced by accepting this H plus ion by the hemoglobin. So this hydrogen ion is accepted by the hemoglobin present in the blood to form what? Hemoglobinic acid. Okay. So now when it reaches the lungs, the hemoglobinic acid under high oxygen tension in the lungs, what happens? There is a plenty of oxygen. So under that conditions, it dissociate and combine with the oxygen to form what? Oxyhemoglobin, and hydrogen ions are released. This newly released hydrogen ions combine with HCO3 minus to form H2CO3. So further it broken down into carbon dioxide and water due to the low tension of carbon dioxide. Clear? So once again, ഇത് മനസ്സിലായില്ലേ? കാർബൺ ഡയോക്സൈഡ് വാട്ടറുമായിട്ട് കമ്പൈൻ ചെയ്തിട്ട് എവിടെയുള്ള വാട്ടറാണ്? RBCനുള്ളിലെ വാട്ടറുമായിട്ട് കമ്പൈൻ ചെയ്തിട്ട് കാർബോണിക് ആസിഡായിട്ട് മാറും. കാർബോണിക് ആസിഡ് പെട്ടെന്ന് തന്നെ ഡിസോസിയേറ്റ് H++HCO3- അയോണായിട്ട് മാറും. ഓക്കെ, അപ്പോൾ ഇവിടെ H+ അയോൺസിൻ്റെ എണ്ണം കൂടുന്ന സമയത്ത് ബ്ലഡിൻ്റെ pH മാറും. pH കുറഞ്ഞു വരും, അസിഡിറ്റി കൂടും. സോ ദാറ്റ്സ് is fatal to the human body. So this H+ ion is received by the hemoglobin to form hemoglobinic acid and transported to the lungs. So in the lungs there is a high concentration of oxygen. So what happens? This HB hemoglobinic acid dissociate, and this HB combined with the oxygen to form what? Oxyhemoglobin is formed. And this H+ ion is released. Now this H+ ion combine with HCO3- ion to form H2CO3. So where it is again dissociated into CO2 plus H2. CO2 plus H2. So this CO2 goes out of the lungs. Okay, so that's how the hemoglobin buffer system works. So that's about what the buffer